Hey everyone, thank you for joining us in Black and Immuno Week, a week where we will celebrate and amplify and support Black voices in immunology. Before we begin, we would like to acknowledge our major sponsor, Immune Deficiency Foundation, a patient advocacy nonprofit organization that helps countless patients in their journey with immune deficiencies. We would also like to acknowledge our major sponsor, Imperial College London, one of the most iconic research institutions in the UK, leading through innovation and passion. You can find all of our contributors on our website, blackandimmuno.org. Today's discussion highlights Black immunologists conducting research in innate immunity, the first line of defense your body uses to protect and eliminate foreign pathogens while working to maintain immune balance. Don't forget to tweet about our innate immunity TEDx talks today using the hashtag #BlackandImmunoInnate, with each word capitalized for accessibility purposes. Our presenters will be using captions with their PowerPoint presentations for our live audience. You can submit questions in our YouTube live feed or on Twitter using our hashtag, again, #BlackandImmunoInnate. Videos will be captioned and posted on our YouTube channel. Transcripts will be made available within 24 hours of the event on our website, blackandimmuno.org. With that, let's get started. We'd like to welcome Antonia Cuff. She is a research postgraduate completing her doctorate at Imperial College London. She studies innate immune responses in pregnancy to help prevent babies from being born too soon. Her work focuses on the type 3 innate lymphoid cells in pregnancy and how they contribute to pregnancy success. She'll be presenting a talk titled "How to Give Babies More Time in Their Mother's Womb to Develop." Antonia, the floor is yours.
Thank you, Dan. Would you help me by sharing the slides, please? Excellent, thank you. Hi, everyone. Thank you so much for the opportunity to talk with you today about my research. Again, I'm Antonia, a research postgraduate at Imperial College London. So let's talk about pregnancy outcomes and giving babies more time in their mother's womb to develop. So I'd like to start off with a story, which at least in the beginning resonates with some of us who might one day want to have children, might one day want to have children, or you might know someone who wants to have children. So this is Nishul and Chandrika. After 10 years of being married, they were excited to find out that they were about to become parents to not just one, but 2 little girls. They were expecting twins. So a typical pregnancy lasts about 40 weeks long. And let's say that on average, that each month has 4.5 weeks. This equates roughly to about 9 months. But halfway into her pregnancy, Chandrika went into labor, and baby Isha was lost at 22 weeks. Baby Ariana was born 6 weeks later, but she needed multiple operations to help her heart and lungs to work. But her tiny body, was unable to cope. And so sadly, after spending months in hospital, Nishul and Chandrika had lost both little girls. Oop. Okay. I wonder, Dan, if it's possible for me to control the slides, because I think the animations aren't coming up quite on time.
When I share my screen, can you upgrade your option to have control of the screen on the sharing settings?
Let's have a look at what I'm being given. No, I don't have control.
Do you, what are your sharing? Do you have sharing options?
Exit image. Let's have a look at this.
I give you. Can you try?
View options. Request remote control. Request. Okay, I seem to have control.
You do? Okay.
Let's give it a go.
Did you try and move the slide?
Yeah. Let's have a look if we can do that. No.
Okay, do you want?
Apologies, everyone. Oh, wait, was that you or me?
That wasn't me. Okay.
Okay, I didn't seem to have that control. My apologies, everyone.
Because it tells me on my screen is waiting for you to control the screen. Okay, you said you are controlling the screen now.
It says I am.
Mm-hmm.
Interesting. Now, okay.
Okay.
I'm moving the, I'm trying to move between the slides, but it's not allowing me to. So okay. I apologize. My computer seems to have gone a little bit funny just before this talk.
Okay.
Okay, let me try and share it with you.
Okay.
And see if that works out better.
So you want to share your screen yourself?
Yes, if that's all right. Let's see if we can do that.
Can you click stop? Okay.
Okay. Okay, are you able to see? Oh dear. Okay, are you able to see the full slides? No, you're seeing my notes.
You want to hide
Why don't we?
view.
I would like to have the presenter view to help, but
I would like to have the presenter view to help, but it's.
I suppose we could try and go without it if that's okay. Swap. Are we okay?
Yes.
Okay. Okay. Yes. Apologies, everyone. A bit of technical issues at the beginning. So let's start again. Okay, so let's talk about pregnancy outcomes and giving babies more time to develop outcomes and giving babies more time to develop in their mother's womb. Okay. So I mentioned Chandrika and Nishul. So they were excited to become parents and they were expecting twins. But halfway through her pregnancy, Chandrika went into labor early and they lost baby Isha at 22 weeks. Baby Ariana was born 6 weeks later, but she needed multiple operations to help her heart and lungs to work, and her tiny body was unable to cope. So when a baby is born before 37 weeks into a pregnancy, they're said to be born preterm or premature. According to the WHO, around 15 million babies are born premature every year, and about 1 million of them die soon after birth. The babies that do survive often have long-term health complications because their body did not get enough time to develop properly. So we need to think of better ways of giving them more time. So one of the things that can cause preterm birth and other pregnancy complications is if the placenta doesn't develop properly. When we begin to understand how the placenta develops in a healthy pregnancy, then we can begin to understand how placental development may fail in some pregnancies and how it can be prevented and treated. So a 40-week pregnancy is often divided into three sections called trimesters, where the lining of the womb goes through a dynamic physical transformation to accommodate the development of a baby. The lining of the womb, known as the endometrium in non-pregnant women, is rich in innate immune cells of the innate immune system. And when the endometrium becomes changed during pregnancy, it becomes known as the decidua. So when an embryo is formed, it must embed in the endometrium to establish the placenta. The placenta is absolutely essential for the success of a pregnancy because it provides the essential nutrients and oxygen the fetus needs to develop. It is formed from interactions between placental cells from the fetus, known as trophoblast cells, and the immune cells in the lining of the womb. So the type of immune cells that you might find in the lining of the womb are mainly innate immune cells, where innate lymphoid cells are the largest population of cells present. So ILC are typically found at mucosal surfaces, such as the gut, the lungs, and the uterus. They're often associated with participating in the immune response to microbes that cause disease and maintaining tissue integrity. There's 5 subsets, which are grouped according to the cytokine proteins they make to carry out their function and the transcription factors required for their development. So natural killer cells, or NK cells, tend to produce the cytokine interferon gamma and require TBET and EOMIS for development. ILC1 also requires TBET for development and produces interferon gamma. ILC2 produces IL4, IL5, IL13, and amphiregulin, and requires GATA3 and ROR alpha for development. ILC3 and LTI both require ROR gamma T and AHR for development, but since they develop from separate pathways, they are considered to be separate ILC subsets. LTI tend to produce lymphotoxins, IL-17 and GM-CSF, while IL-22 is the main product of ILC3. So we know the role of some of these ILC during pregnancy, but we do not know the role of all of them. So NK cells are the predominant ILC in the uterus, and they contain a subset that resembles ILC1, but it's really not clear whether uterine NK cells are actually NK cells or ILC1. But nevertheless, they have a known role in promoting placental development. ILC2, on the other hand, are rare in the decidua, although few studies have looked for them specifically. And finally, there are the ILC3 and LTI cells. So in the past, mouse ILC3 subsets were thought to include the LTI cells and ILC3. However, more recently, LTI cells have been found to develop from a separate progenitor to the ILC3 and have therefore been reclassified as a separate ILC subset. One of the things that contributed to this overlap was because most of what we know about how ILC develop and what they do come from studies done in mice. And when scientists try to identify them in humans, they may not always produce the same proteins as we've seen before in mice, and so they may look different. More recently, a number of studies have identified ILC3 in the lining of the womb in humans, particularly during the stage when the placenta begins to develop. And although we know that these ILC3 can produce IL-22, we know little actually about what they do at this stage, and indeed throughout the rest of pregnancy. But what we do know is that mice lacking the IL-22 protein are susceptible to bacterial infection and disintegration of the structural column of epithelial cells that lines the mucosal tissues. So this highlights the important role of IL-22 in maintaining the epithelium, as well as providing protection against microbes. We also know that IL-22 protects against fetal loss in an infection model of pregnancy. So the gray mice can still have babies in the presence of an infection, while the white mice will lose their babies, unless they're given supplements of IL-22, which rescues this fetal loss. And the way IL-22 works is by promoting the survival of epithelial cells and a rapid expansion in their numbers. So it's possible that IL-22 may be acting the same way in the uterus. And in the context of pregnancy, the placental trophoblast cells from the fetus are epithelial-like cells. And so for my PhD, I've decided to ask the following questions. How does ILC3-derived IL-22 contribute to establishing the placenta? And how does ILC3-derived IL-22 maintain the lining of the womb to allow a fetus to develop to term? I begin to answer these questions by screening uterine samples from different stages across pregnancy, including the stage before pregnancy happens in non-pregnant women. And then I ask these sort of questions from the data. How many ILC3 are there? Which cytokines are the ILC3 making and how many? And does this change during pregnancy complications? But before I can do any of this, I need to be able to identify the ILC3 cells. So in the past, all studies often lacked the tools to examine ILC3 separately from the other ILC subsets that might look similar to it, like the LTI cells, for example. And this made it difficult to determine the unique contribution of the ILC3 within pregnancy. Nowadays, we have better research tools to identify human ILC3 separately from one another, which makes finding out what they do a little bit easier. So one of the first things that I did as a first-year PhD student, without access to the lab in the first lockdown, was to do an analysis that would allow me to identify ILC3 in uterine samples. So the data I'm showing you here is from a single uterine sample from a first trimester pregnancy. This analysis was created using what is known as a high dimensional analysis, and it's an analysis which allows me to visualize multi-parameter data in a smaller two-dimensional space, which is easier for the human mind to grasp. So in addition to reducing this data into a 2D format, I also did an analysis on the same data to create clusters of cells that are similar based on their expression of similar proteins. And so using this information, I know that, for example, that the orange, the lavender, and the green cells are NK cells, and the blue cells are the ILC3 that I'm interested in. So once I'm now equipped with the best strategy to identify the ILC3, I can use this on actual uterine samples in my analyses. Okay. So since the lab reopened after the first lockdown, I've had the opportunity to screen for uterine ILC3 in endometrial, first trimester, and third trimester tissue. And I found that uterine ILC3 are most numerous during early pregnancy when the placenta begins to develop, similar to what is already known. So this suggests that this is the key stage where their activity is most likely to be important. And so I've designed experiments to analyze the effect of ILC3-derived cytokines on their most likely cell targets, the fetal placental trophoblast cells, which are the major drivers of placental development. I will compare this data to uterine samples taken from women who kindly donated their samples, who are experiencing pregnancy complications, in order to find out if anything that the ILC3 are doing contributes to or helps the complication. So the WHO says that the largest barrier to the development of diagnostic, treatment, and prevention strategies for preterm birth and stillbirth is our inability to comprehend the biological processes of pregnancy and childbirth. So that's where reproductive immunologists like myself and the clinicians like whom I work with come in. We are trying to understand the biological processes of pregnancy and childbirth so that we can create better diagnostic, treatment, and prevention strategies for preterm birth and other pregnancy complications. So remember when I told you the story about Nishul and Chandrika at the beginning? Well, I'd like to end my talk on a positive note to tell you that, with help from the team at the Bowen Charity, who funds research or conducts research on ways we can prevent preterm birth, they were able to have a third child, baby Ahana. She was born just shy of the premature birth cutoff mark at 36 weeks. But luckily for her, unlike her siblings before her, she did not require intensive care. But so many families remain affected around the world every year, and preterm birth rates are not decreasing. So if we can imagine the scenario where uterine RC3 contributes to the biological processes that allow the placenta to be established, and the processes that allow the lining of the womb to be maintained to term, then we can begin to understand if uterine RC3 are affected in pregnancy complications like preterm birth. And if they are affected, then we may also begin to think of ways we might be able to treat or prevent those complications, giving babies more time in their mother's womb to develop. And so that's the challenge of my PhD. And this is the team that I work with that enable this work to be possible. Thank you all for listening. And I apologize for the technical error at the beginning. Thank you.
Thank you, Antonia, for your presentation on RC3s and pregnancy. And we again encourage our audience to submit questions now for our Q&A discussion that will follow these presentations. Next up, we have Dr. Dabrowski Herbert. Dr. Herbert is an associate professor at the University of Pennsylvania in the School of Veterinary Medicine. The Herbert Lab explores the cellular and molecular regulatory networks controlling inflammation that drives host protective immunity and tissue repair in humans and rodents infected with parasites. This research program is particularly focused on parasitic helminths, which is our major cause of morbidity in human populations and agricultural animals across the globe. And today, Dr. Herbert will be presenting on overcoming barriers, pushing limits, and dispelling the imposter myth. Dr. Herbert, the floor is yours.
Okay, can everyone hear me okay?
Yes, we can.
Okay, great. Thank you, Daniel, Elaine, and Justine for inviting me. It's quite an honor to participate today. I've prepared a short presentation for you along in the spirit of the TED Talk format. I wanted to literally infect those who are listening today with a couple of ideas. The first idea, and I think prevailing throughout, is really, if you look on a global scale, how we are sitting on one home, you know, and while we are very diverse Homo sapiens, we certainly have various different perspectives and talents. And, you know, as a scientist, as an immunologist, I've been asking myself for quite a long time, who am I? What is my place in this large, confusing, complex world? And how can I leave it to be better off than when I first started or when I entered it? And I think, you know, being an immunologist really encompasses all of that. And actually through my training and through developing as a professional, I've been able to even, you know, travel the world. And this story for me, it really is the African-American experience, and that you can see from my genealogy here that I'm predominantly Nigerian, and I am proudly wearing my Nigerian shirt today that was made for me during a collaborative visit in Lagos. But you can see here, there are many other countries involved in my genealogy. And I think that, again, highlights how, you know, difference in perspective, but also the commonality in Homo sapiens is very important. And so, as Daniel pointed out, yes, I have very scientific interests, primarily in the area of parasitic helminths, but that has taken me into focusing on allergic diseases and wound healing responses. So type 2 immunity is generally what my laboratory is known for. And for those of you who are not familiar, it is a prototypical immune response characterized by, excuse me, a variety of interleukins and cell types. And as you can see here, interleukin 33, which I will tell you quite a bit about today, IL-25, TSLP, and a whole host of characteristic cell types that over the years my laboratory has studied. And I must say from when I first started in early 2000,
Dr. Herbert, just to let you know, if you're meant to be sharing a screen right now, we're not seeing anything. Can you share your screen?
Can you see that now?
Yes, if you go ahead and present the slideshow and we'll be good to go.
All right. All right. Well, I'm going to just quickly go back here and say, you know, that this worldview is very important for a basic scientist, and, you know, being Nigerian for me helps not only to solidify my worldview, but also give me some sense of ownership and pride in who I am today. And so these are the types of settings that we study in the laboratory, as I pointed out, in parasitic infections, which disproportionately affect billions of people across the globe who are underdeveloped countries, I would say. And allergens and wound healing. These are all topics that we study in the lab, and they're all largely under the umbrella of type 2 immune responses, as I was stating a few seconds ago, is a type of response that's initiated by barrier damage and has different key cytokines that accompany this type of response. And over the years, we've focused on helper T cells, ILC2s, and alternatively activated macrophages. And so, you know, when along these lines of breaking down barriers and overcoming obstacles, you know, many times in my career, I've thought about, you know, what choice am I going to make? What path am I going to follow? And here's an example of two paths. And I most certainly have found over the years that I definitely like the path less traveled. And so, you know, building on this idea of taking paths and building your career, one thing can perhaps use the analogy of building a road that has many different components to it. I mean, we're certainly annoyed at sometimes when we have to take detours when construction is ongoing. But my question or my challenge for you today is to think about what participant or what kind of vehicle for preparing that road are you? Are you in the early stages? Are you a front loader? Or are you a steamroller? In essence, you know, to really pack down the asphalt and fill in the small crevices? Or are you this svelte, you know, Tesla, right? That's just kind of silently going over the road, you know. And I must say that I realized today that I'm very much a front loader. I've always been ambitious and not cowed to go into areas of science that are less traveled. And I'm going to provide for you a couple of examples of that. During my postdoctoral days, I went against the grain and went to South Africa when most of my classmates were going to NIH and other high-profile laboratories in the US. I was adamant that going to South Africa, that was made possible for me to do research there by this individual here that has no need for introduction. Nelson Mandela really did a remarkable thing for the globe, not only for the country of South Africa, and made it possible for me to publish this paper that formed the basis of really my identity as a scientist in these early days. And it formed my basis for my first R01 and the basis of my early laboratory. And it was really born out of a lot of hard work and drive to do so. So M2 macrophages, this was one of the very first papers that solidified the importance of this cell type in the field. And I moved on to Ohio, where then, you know, training with mentors is certainly important. And, you know, there's a lot of kind of anecdotal evidence out there that the individuals you train with really make a indelible impression upon you as a scientist. And I would say that Fred was quite a maverick in his own right in studying interleukin 4 and you know, kind of solidifying those pathways and working with Fred. I struck out on my own and still working on parasites. We published some nice work indicating that there were soluble proteins released from epithelial cells that actually eliminated parasites by preventing their ability to find food and to feed. You can see here that molecules realm beta, which exposure caused those parasites to become blanched or clear. Whereas its very closely related molecule, realm alpha, was unable to interfere with worm nutrition. And you can see here the kind of magenta-like hue of this parasite that is a blood-feeding parasite. So we did a lot of cool stuff together, but again, I want to charge you with the idea of making your mark. So it took me a while, but I finally formed the thesis that I would still hold today as really the driving force for the laboratory. And that is that mammalian coevolutionary adaptation to helminths has driven tissue repair to become immunoregulatory. And once I formulated that statement, that shaped everything else. Then going on for, using some of the basic metrics of organizations like the Burroughs Wellcome, which I'm a fellow of, I set out for success. That is to build my team, form strategic collaborations, establish my own niche in the field, and of course, which I put a little icing on the top was to form a lab mascot, and that is Horatio. And everybody's pretty happy, at least at that point, for working with Horatio. And so as my own team member, I decided to focus on respiratory diseases. And I'm not going to go through this slide in any great detail for in the sake of time, but there's certainly leading causes of morbidity and mortality across the globe. And perhaps this also needs no introduction that now we're still in the midst of this global pandemic. So what was my take on the matter? What was my particular contribution to the field? Well, that was to use hookworms to understand respiratory disease. And in fact, we found that parasites were causing these particular areas of injury, but they were being very rapidly resolved. So here's the model system in which to ask the question. And here we established some techniques that such as pulse oximetries, that if anybody went into the emergency room, this is what the first thing they would do was test your blood oxygen content. So we established this for mice, and off we went. And I must say that I remember it was during an immunology retreat. I knew I was looking for a molecule. I think serendipity is something that is granted to the person who is looking for something in particular. They just don't know exactly what it is. But when I found when one rogue presentation, the presenter talked about a repair molecule that they didn't have a mechanism for, but it was called a trefoil factor. I knew at that moment that all the lines, all the ducks were in a row, if you will, and that this is what my lab was going to study. And so trefoil factors are still not well understood. We have lots of funding and papers on the matter, and so it's rife with areas of opportunity, but these are molecules that are produced by damaged epithelial cells and other cell types at the border of wounds, and they aid in this restitution. They aid in the ability of these wounds to close. But now, of course, the immunology wasn't known. So we developed our own systems, our own fluorescent reporter conditional mutant mice, and found that actually in the lungs of mice that were infected with hookworms, trefoil factor 2 expression, shown here as red fluorescence protein, really demarcated those particular areas of injury. And so, we published several pieces of work along these lines, built on this model and this molecule system. And this is one of them here, in that we link trefoil factor 2 with interleukin 3 signaling in the context of both allergic asthma and hookworm infection. And again, another thing I really want to emphasize here is the importance of building models. And I often instruct or encourage rather my trainees to get a sketch pad and draw out what they're thinking to solidify and hone those experiments. And this is one example of that. You can see the papers that this model is supported by. But then went on to California, to UCSF, and it was there, that's UCSF is really these top tier research institutions. Some people call it colloquially a shark tank. And I knew we had to take on big questions in order to survive to swim with the sharks. one of the biggest questions in the trefoil factor biology field was, is there a receptor? And even though I'm a cellular immunologist, I turned myself into a biochemist, and we used this very kind of a radical approach at the time to identify trefoil factor binding proteins on the surface of cells. And we went on to use this approach, it's called triceps, to publish this paper last year. Actually, it finally came out, indicating that LINGO-2 was a receptor for trefoil factor 3. So this has opened up a whole new area of research in the lab and a considerable amount of funding. And lastly, kind of coming home, I will say, in some stretch of the imagination, to Philadelphia, we've gone on to continue to focus on interleukin 33 and to break down another barrier, and that is, this prevailing idea that interleukin 33 is a molecule that's released from damaged or dying epithelial cells, and then once it's elaborated into the extracellular environment, it can then act on a variety of different innate and adaptive immune cell types to augment type 1, type 2, type 17 immunity, even T regulatory responses. But there's been a lot of question about, is this paradigm exactly correct? And in fact, we posed two questions very recently, I would say over the past 2 years. Could myeloid cells be important sources of IL-33? And also, was there a specific mechanism of release other than the cell death pathway? And I'm very happy to say last week, we finally got this paper out in Science Immunology. And indeed, this is going to change the paradigm of how we think about IL-33. It was a hard road. It took us many years to get it, but we have it now. And we identified a new secretion pathway for interleukin 33 that may be relevant for a whole host of other settings outside of parasitic worm infection. So I encourage you to take a look. And so, again, I'm going to leave you with this working model, and that what the paper is really trying to put out in the field is that, in contrary to the established paradigm where IL-33 is a nuclear resident cytokine of epithelial cell types or barrier type cells, that it gets elaborated upon the death of these cells, that it can expand innate lymphoid cell populations like ILC2s. We identify that antigen presenting cells such as mucosal dendritic cell subsets, not only do they express IL-33 in a different compartment, so they express it in the cytoplasm and not so much in the nucleus versus these cells, but they actually encode a pore forming protein that is regulated by IL-33, and that pore forming protein facilitates IL-33 delivery to T regulatory cells, a very distinct subset of T regulatory cells, the ST2 expressing Treg subset, that then can come back and suppress this inflammation. So this is almost a self-closing loop that we've identified in this work, and I certainly hope that many will follow this and with other studies. And so the field is, my group rather, the field also has grown, but my group has certainly grown over the years. This is an outdated picture. It doesn't have Yvonne and Jay and Juan and all these really bright individuals who have decided to join the team, and I'm so grateful for. But I want to leave you with a thought. And that is, to my sharpest minds and those considered not so bright, to my people of the lightest hue and those that can pass for night, let your light shine and do not apologize for being uniquely you. Assimilate only in excellence, but in nothing else that you do. And when they try to make it seem that you are a source for sympathy, know that Black Tax has built your stamina to reach infinity. When there seems not enough known or no reagents to test that condition, know the only block to progress is your willingness and volition. Since the middle passage, thriving against all odds, is our specialty, even using past atrocities as brick and mortar to shield against any mental instability. So stand tall, Black in Immuno, speak confident and clear, pipette with precision. Your contribution to humanity awaits sharing your perspective. Just make your decision. Thank you very much.
Dr. Herbert, thank you so much. Thank you for sharing your work on helminth infection, the path that you paved, as well as those words of encouragement at the end. Thank you. Again, we encourage our audience to submit questions now for our Q&A discussion that will follow all of our speakers giving their talks. So next, we will now move to Dr. Bernice Mbiri-Bindi. She's a senior postdoctoral research fellow at Stanford University School of Medicine and Professor Sherry Cram's lab, focusing on transplant immunology. Her research interest focuses on NK cells and the impact of the major histocompatibility complexes class 1 and modulating their functions. Combining in silico and in vitro analysis, she's working to understand the role of NK cells in the context of Epstein Barr virus latent infection and their involvement in EBV-related complications. Dr. Mbiri-Bindi is joining Genentech later this year, and her research will focus on immunotherapy. Today, her talk is entitled The Impact of Viral Peptides on Natural Killer Cell Function. Dr. Mbiri-Bindi, the floor is yours. It sounds like you might be muted right now, so go ahead and try and unmute, please.
I'm sorry about that. Thank you very much, Dan, for this introduction. Hi, everyone. I want to say that I'm extremely excited to be here, and I'm looking forward to the amazing week ahead. My name is Bernice Mbiri-Bindi, and I was a postdoctoral research fellow in Sherry Cram's lab at Stanford. And as Dan mentioned, today I will be talking about the impact of viral peptide on NK cell function. So I hope we can all agree that when I hope that we can all agree when I say that the immune system can be described as a powerful army that protects us from viral threats. This personal army patrols our body and is ready to fight any attack. And there is 2 ranks within that army. The first line of defense, also known as the innate immunity, and the specialized line of defense, also known as the adaptive immunity. And among the innate human cells, we found those amazing and powerful cells called natural killer cells. Natural killer cells, well, it's really all in their name, right? They are the first line of defense in the battle against viruses and cancer. They have the ability to rapidly seek out and destroy abnormal cells. Natural killer cells have 2 types of receptors: activating receptors that put them to work, and inhibitory receptors that prevent them from attacking healthy cells. When scanning our cells for their integrity, natural killer cells look for a protein produced by normal cells called the major histocompatibility complex class 1, or MHC1 for a more digestible name. And NK cells also pay great attention to the peptide that those MHC molecules carry. Peptides are short pieces of protein that basically help sample what's going on in each of our cells. We can look at it this way. MHC class 1 molecules are like flags that's at the surface of our cells that wave at the immune system to tell them what's going on. And peptides are the design within that flag. And it basically says what type of things are happening within the cell. So as the title of this talk mentioned, I will be talking about how NK cells respond to viruses. And I have to say that viruses are really smart. After infecting their host cell, they make it their mission to evade the immune system. They play hide and seek, and they put a lot of effort for that. When it comes to NK cells, some viruses use that flag system we just talked about. They can make the infected cells engage with the inhibitory receptors on NK cells and effectively put them to sleep. Viruses will often change that design within the flag to trick NK cells. This can benefit them, and in the long term, they can also establish chronic infection, as some viruses do. But sometimes that trick doesn't really work, and it pushes NK cells to attack and eliminate the infected cell. Our lab at Stanford studies Epstein Barr virus, or EBV for short. We study the infection as well as pathologies driven by this virus. I need to mention that this virus is our friend for life. In fact, the majority of the world population is infected by it, but it doesn't really cause a problem because most of us are immunocompetent and we can fight it and keep it in check. However, This virus has the potential to cause cancer among people who can't fight it and is linked to several forms of diseases worldwide. People who are immunocompromised, people who are immunosuppressed, such as transplant patients, for instance. Studies in our lab, but also others, have led to two key observations. One, EBV infection increased the expression of those flags we just talked about earlier, the MHC class I molecule. And two, there is a specific population of natural killer cells that can react against these infected cells and attack them. Then we asked, what if during the infection, the virus was generating peptides that the NK cells could not recognize, which will then push them to attack the infected cell? In this project, we looked at the interaction between HLA-E molecule, which is an MHC class I molecule, and NKG2A, which is an NK receptor, since the cells that were responding to EBV infected cells were expressing that receptor. I won't say that NKG2A is normally an inhibitory receptor, so it kind of, when it recognizes its ligand, so the HLA-E, it puts NK cells to sleep. But here we're seeing that NK cell that expresses NKG2A are getting triggered to EBV infected B cells. So we use this cool system that allows NK biologists to study the interaction of a specific NK inhibitory receptor with its ligand, the MHC class I peptide complex. I want to take a few seconds here to describe this reductionist model. So basically earlier during this talk, I mentioned that healthy cells display MHC class I molecule at their cell surface, like flags that allow the immune system to know what's going on in the cell. And I also said that those flags have design, so the peptide, that were generated within the cell. And all of this happens thanks to the cooperation of different machineries within the cell. What we can do as researchers is to hijack that machinery and cooperation to stop that process to happen, which will then allow us to literally feed the cells with our peptide of interest and have at the cell surface an overwhelming expression of a single MHC class I molecule. We can basically then be able to study each individual peptide and how they affect a given NK cell population. So for this project, we're investigating HLA-E interaction with NKG2A. With this system, we can do two things. One, we can measure the increased expression of HLA-E level at the cell surface after feeding the target cell with individual peptide, meaning that we can identify which ones are able to be carried by this molecule. Two, we can assess how the MHC class I peptide complex influences NK cell responses. So for this work, we used a prediction algorithm to predict potential peptide from EBV proteins that can be shown at the cell surface by our MHC1 molecule of interest. We had a long list of peptides and we tested them through that reductionist model I just talked about. Here, I'm just showing one sample data of two proteins. And the first question that we asked was to know if biologically our MHC class I molecule, so HLA-E, were able to carry those peptides. I've highlighted in black the one that was selected as being binders to HLA-E. And I want to say that we also used peptide controls that are shown at the end of this graph that are well studied and well known in the literature to bind to this MHC molecule. Secondly, we looked at the ability of those different peptides to either inhibit or activate NK cells. Here, I'm showing a sample group of data, and I wanted to show you that we again have those control peptides that are known to drive different effects on NK cells. So one of them to inhibit NK cells, while the other one releases NK cells from their inhibition. And we can see that within our library, we're able to identify peptides that was actually were able to trigger NK cell activation. Lastly, we looked at the ability of NK cells to kill target cells that were harboring those peptides. You can see here, that the target cells are being killed, and this is depicted by an increased level of cytotoxicity. So after combining all these results, we wonder what these NK cells were seeing when they were scanning those target cells, and how HLA-E was interacting with NKG2A to abrogate its inhibition. So there is a collection of data and structures explaining how those two entities interact and when they see each other. And we know that there is two residues within the peptide sequence. So the peptide is here. And there is two residues that are key for that interaction, as highlighted here. These residues can be found at position 5 and position 8 of the peptide sequence. Their conformation are really important for the inhibition through NKG2A receptor and essential for the association between NKG2A and its co-receptor CD94, which will then lead to NK cell inhibition. So we decided to model our peptides in order to analyze the conformation of those residues. In this busy slides, I'm displaying a couple of peptides from our EBV library. So peptide 12 on the left and peptide 29 on the right. I also added the controls that was used during those experiments. And we know that peptide 12 and 29 have two different effects on NK cells. And I'm also highlighting here at position 5 and position 8, those important residues. So I want to drive your attention to the conformation of these residues. I want to drive your attention to the conformation that these residues are adopting. We observed that residues at position 5 and 8 of these two groups of peptide have different conformation. And in a nutshell, basically, peptides that allow NK cell activation do not tend to favor the association of NKG2A and CD94. And we observed the opposite for peptide that did inhibit NK cells. So I hope that you have been convinced this morning that, this morning in California, that with the combination of in silico and in vitro analysis, we're able to show that EBV proteins can generate peptide that can be presented by MHC class 1 molecule and trigger NK cell activation, resulting in target cell killing. Furthermore, we also added some modeling data that suggests that those peptides are actually impairing the recognition of NKG2 and CD94 heterodimer, which then lead to NK cell killing activity. Additional details of this work, and there is more, can be found in our recently published paper. And I want to finish by saying that I believe that science that focuses on understanding mechanism by which the immune system can overcome tricks from viruses will better our knowledge on these viral infections and also allow us to understand how to control them. So now when you think about NK cell receptors and MHC class 1 interaction, I hope you will also now start thinking about the peptide as well. With that, I would like to thank my former lab at Stanford, the Transplant Immunology Lab, my mentor, Sherry Krantz, as well as everyone involved really closely to this project. Obviously, I also want to say thank you to the body funding, the funding bodies, and thank you for your attention, and thank you very much for tuning in.
Thank you for that great talk. That was really great work, and I really appreciate you sharing the work that you've done and recently published on these viral peptides as it relates to EBV. Again, we encourage all of our audience to submit questions either through Twitter or on the comment area on the YouTube Live website. So next, we will turn to our next and final speaker, Dr. Juliette Morrison. Dr. Juliette Morrison is a virologist and assistant professor at the University of California, Riverside. She earned her Ph.D. in microbiology at Columbia University, completed her postdoc training at Mount Sinai School of Medicine at the University of Washington. Dr. Morrison currently researches host responses to human viruses, including dengue virus, yellow fever virus, and the influenza virus. Although host responses have been implicated in viral diseases, current therapeutic interventions generally target only the viral replication. Her goal is to translate her findings into host-targeted therapeutics for viral diseases. The title of her talk today is Innate Immune Drivers of Influenza Severity. Dr. Morrison, the floor is yours.
Thank you. And thank you to the organizers for inviting me to give this talk. I'm very excited to be here. So today I'll be talking about influenza. And influenza is a very important human pathogen. It has been around forever. It was first described in the early 400, as early as 400 BC. And it has been the cause of major epidemics and pandemics throughout history. One such pandemic is the 1918 influenza pandemic. Here I'm showing you a photo from a hospital during the great influenza pandemic. And interestingly enough, that pandemic was so, induced so much mortality that it killed more people than World War I. And many experts believe that another influenza pandemic could kill millions and that this is actually inevitable. So seasonal influenza is quite a burden. It results in 3 to 5 million cases of severe disease and up to 500,000 deaths annually. It costs the US approximately $11 billion per year in costs related to influenza. And despite the availability of vaccines and antiviral drugs, influenza is still common. And that is because influenza mutates rapidly. And as such, drug resistance is a major concern. Many of the drugs that were created to target the virus are now actually obsolete. But you know what doesn't mutate easily? Us. And that's why I'm interested in host-targeted viral therapeutics. So disease severity is determined by the interplay between the virus and the host response. And basically the host response is just the reaction of a living system, like our bodies, to a stimulus. Too little of a response is bad because then your body would not be fighting the virus, but too much of a response is also bad because this can lead to excessive tissue damage. And so one of the goals of my research is developing host-targeted viral therapeutics. And the way I see this is as a seesaw with viruses on one side and host responses on another side. And we want this seesaw to be level. And if you look here, you can see that the viruses are currently winning. But if we were to just move this little responder a little bit over, then we could actually see balance. And that is one of the things that I am trying to do in my research. And so we are trying to identify pathways and cell types that we can target to improve influenza outcomes. And to do this, we take an approach of analyzing gene expression data, modeling this data, and then doing experimental validation. So one of the questions we've asked was, is there a long gene expression signature that can distinguish between mild and severe influenza? And we did this by comparing the host response to different influenza viruses. So here are all the virus strains that are involved in this study. And it's actually from 2 different studies that we've combined. And if you look here on this graph, we have percent body weight on the y-axis, and on the x-axis, we have the days post-infection. And you can see that there are a variety of outcomes with the different influenza virus strains. So here with milder strains of seasonal influenza, we see a little bit of weight loss and then recovery. In some situations, the disease is far more severe with a lot of weight loss, and eventually the animals die. So what we did was to get lungs from these animals and then to look at the viral titers, gene expression, histopathology, and with the ultimate aim of identifying tissue signatures that could distinguish between the mild and the severe influenza. So here are our range of disease outcomes. And we also have here some gene expression data. And I want to point you to the percent weight loss here, where I've lined up the virus infections from mild on the left side to most severe on the right side, most severe including this 1918 influenza here. And I want to bring your attention to one of these modules, gene expression modules. This orange module here that is where you see higher expression of lipid metabolism and coagulation signaling genes in mild outcomes and less of the expression in severe outcomes. So pathogenic viruses induce a transcriptomic signature of decreased coagulation and lipid metabolism. But what is actually driving this? Which lung cells are responsible for these transcriptomic differences? Because we know that the lung is not just one homogeneous tissue. It has epithelial cells, multiple immune cells, different endothelial cells. So it's a complex tissue. And so to parse this out, we took a bioinformatic approach and we used a program called Digital Cell Quantifier, or DCQ. And what DCQ does is to take genome-wide gene expression data, like the one we have, and combine it with gene expression data of 207 different immune cell types. And this leads the algorithm to infer the in vivo global dynamic changes in immune cell quantities within complex tissues. And as such, DCQ can predict the temporal process of immune cell activation in a mouse model. So we did the DCQ and then we identified populations that correlated with weight loss, and weight loss is our readout for disease severity. Here I have them lined up again from least severe to most severe. And DCQ predicted that dendritic cells would decrease with severe influenza and that neutrophils would increase. And this is actually already known and published in the literature. So this was just a confirmation that the algorithm works. It also predicted other known phenomena, such as the decrease in alveolar macrophages and the increase in monocyte populations during severe disease. But we weren't trying to use this algorithm to tell us what we already know. We were using it as a discovery tool. And so I'd like to point you to this other population here, small peritoneal macrophages. So what are peritoneal macrophages doing in the lung, and what even are peritoneal macrophages? Well, this is where the story gets exciting. So we, mammals, we have these things called serosal cavities, and what they are fluid-filled cushions, basically, around our organs. So the serosal cavity that protects our abdominal organs is called a peritoneal cavity. And the one that protects our lungs and cushions our lungs is called the pleural cavity. And since these serosal cavities are analogous, they also contain similar cell populations. And so they can be divided into two populations of macrophages, small macrophages and large macrophages. And they're distinguished by both their size as well as the markers on their surface. And so what our hypothesis was then is that it's not actually small peritoneal macrophages, but perhaps small pleural macrophages that are going to the lung. And so we hypothesized that these small pleural macrophages, and perhaps even the large ones, can cross the mesothelial layer and enter the lung, where it can help to resolve infection in some way. So we have to figure out if we could actually label our pleural macrophages so we could track them. And to do this, we do an intrapleural injection with a macrophage dye called PKH. And so macrophages take this up and they fluoresce red. So when you harvest the pleural cells, you can see that the macrophages here are fluorescing red. So we are able to label these macrophages in vivo. Also, I want to point out that we have different, we have two different viruses, California 09 and PR8, which cause mild disease. So here we're looking at a lung, and PR8 causes severe disease. So you see all of this debris in the lungs, etc. And this is PBS, which is our control. So we have that set up, and we inject our mice with the intrapleurally with the macrophage dye. And then the next day we do an infection with the Cal09 to cause mild disease, or the PR8 to cause severe disease, or we have PBS as our control. Then we harvest these lungs, And you can see here that in the PBS treated animals, the lung tissue has very little red in it, indicating that the pleural macrophages have not entered the tissue. However, the California 09 infected mice have a whole bunch of red in the tissue. The PR8 infected mice, which go on to die, they have less of these macrophages. And so we are seeing this accumulation of the PKH positive cells in the animals with mild disease. And so these findings support our initial prediction, which was that we would see macrophages, pleural macrophages even, in animals that with mild disease, and that it would be absent in the animals that develop severe disease. And so I've shown you that we have used whole lung transcriptomic data and bioinformatic tools to identify a novel cell type that could impact influenza outcomes. And our data so far suggests that the pleural macrophages migrate to the lung during influenza virus infection. So that means we actually have a whole bunch of questions that we'll now be able to follow up on, such as what's driving the macrophages across the mesothelial layer. What are they doing once they actually go to the lung? Are we seeing decreases in the viral load, or are we seeing impacts on cytokine levels, etc.? And I believe that targeting pleural macrophages could be a promising strategy for treating influenza and other lung infections. So finally, I'd like to acknowledge the people who have made this possible, especially my graduate student, James Stumpf, whose project this is. And thank you. I'll take any questions during the question and answer session.
Dr. Morrison, thank you so much for sharing your work. That was fantastic. It was great to hear about everything that you're doing. And so now we'll be transitioning to our Q&A period, and we'll be opening it up to all of our panelists. And so for those who have tuned in the middle of this, our speakers for today are Antonia Cuff, who is looking at innate lymphoid cells in pregnancy. We have Dr. Herbert, who is looking at Helminth infection, as well as the path of his career that led him to where he is now. We have Dr. Bernice Mberibindi, who's looking at EBV viral peptides and their role in NK cell activation. And Dr. Juliette Morrison, who just finished her talk looking at health outcomes for various influenza viruses. And so to begin our Q&A panel, I just wanted to open up the floor to all of our panelists and say that I really enjoyed hearing about all the work that all of you are doing. It's fascinating work. And I wanted to know what is the passion behind your work? What motivates or animates you with the work that you're doing? Dr. Mberibindi, if you could start us off, that'd be great.
Oh, thank you, Dan. For me, the interest of natural killer cells started when I was finishing my master's degree. I used to focus on T cells before and B cells, and then I heard about natural killer cells, and I was so fascinated by the way they works and the way they actually act so rapidly and make tiny distinction between something that is normal and something that is not normal. And get rid of, infected cells or cancer cells. And the reason why I'm kind of trying to stay on this track is because I feel like there is a lot of things to discover regarding NK cells, and they're kind of like these misunderstood cell types. And as Antonia pointed out in her talk, you can see that there is also cells that are being discovered that look like them, but not quite. And yeah, so there is still a lot to uncover, and I think that's why those cells are really fascinating for me. Thanks.
Dan, are you going to call on us sequentially, or you want us to just jump in? How do you want to do this?
For anyone who has an answer, go ahead and jump in. So Dr. Herbert, if you have an answer, go ahead, please.
Okay, so this really speaks to my childhood, I would say, and during undergrad. So they'll give you two examples. One was, as I was growing up, my great-grandmother told me to not go outside without my shoes on. And, I grew up in the deep South, and I really couldn't get my head around why couldn't I go outside without my shoes on. And, it was early in college I figured out that my grandmother was warning me about hookworms. And so with a scientific mind, I just wanted to understand more and more about the unique biology of these organisms. And that's really where it came from. And the more I learned, the more interested I became. And when I went to UCSF during undergrad research, one of my first projects was to work on CD8 T cells in the context of malignant melanoma. And I realized as someone with as much melanin in my skin as me has a less likelihood of developing malignant melanoma. And I just felt that if I was going to work as hard as I was working, that I wanted to give my life's work to diseases that would affect disproportionately people from developing worlds. That's why I do what I do.
For me, oh, go ahead. Sorry, you go ahead. You ready?
No, go ahead.
Oh, okay. For me, I really enjoy the work that I do. I think it didn't start out as great as it is right now, as in the fact that I feel like the work that I do has meaning and that it has potential to affect human life in the best way possible. So that's a motivator. But the day-to-day, I do enjoy learning and studying the immune system. I have sort of moved between different immune cells since I've started in science. Beginning learning a little bit about ILC2s as a research assistant in my early years in Cambridge, and then more getting to understand the immune response as a research assistant again, looking at the immune response in transplant immunology. And then now I've moved on to so ILC3 from the NK cells, but still focusing within the ILCs. So the immune system has always fascinated me, and now that I get to study and learn all the time, which I do enjoy doing, learning and studying, that's what I enjoy and keep going and hope to keep doing as long as I can, really. So that's for me.
I do this because I really like asking questions and answering them. Specifically, I'm actually a virologist by training, and it was some years ago that I remember reading a paper about dengue, which is one of the viruses I study. And it said that the severe dengue actually occurs after the body has already cleared the virus. And that just stuck with me as something that wow, okay, we put all this, like we think viruses are so important and that they're the big drivers of disease. But in the case of severe dengue, it's actually the host response to the virus, because at that point, the virus has already cleared. And that got me very interested in just host responses in general. And so in the past few years, I've been really trying to learn more about innate immunity. And that's what we're now focusing on in my lab, which I started 2 years ago.
So now to try and turn a little bit more towards some scientific questions. One of the emerging questions in from our listeners is for you, Antonia. They were wondering, do you know if the IL-22 knockout mice reproduce okay in your system?
Okay, that's a really good question. So I actually don't have these mice myself. This is based on a study which is published in a paper like maybe a year or two ago. So they reported that the number of fetuses as a percentage of fetuses that survive. So I'm not sure if, for example, the numbers are smaller in IL-22 knockout mice compared to wild type mice, or if it's just that the number that were born, they survive well in the absence of an infection. So that would be something interesting actually to look at.
Okay. And Bernice, one question that kept emerging for you was, do you have any thoughts on how your work on EBV might represent the emerging concept of innate memory?
Innate memory and NK cells, yeah. So my work doesn't really focus on that in that aspect, but it's true that for the work in memory NK cells, there is this big question mark about what type of antigens are driving that. There is some that was found with the human CMV virus and KG2C receptor, but this is a total different type of mechanism. But I know that there is still a lot of questions about what type of antigens, so peptide, are driving the arisal of memory NK cells. But yeah, I mean, yeah, my work is kind of slightly different from that.
Okay. Well, how about from the viral peptides that you were exploring in your work? Do you know that if these viral peptides are would they emerge in the context of other viral infections like cytomegalovirus, or is this a unique mechanism to EBV?
Well, those peptides are unique to EBV since they are basically encoded by, they are derived from EBV proteins. So something that I didn't really mention in the talk because of the sake of time, it was that in our body, the proteasome, which is one of the machinery I was talking about, cuts our proteins into small pieces, and those are called peptides. And they do the same thing with viral protein as well. And that's how we basically identify those peptides. They don't have any overlapping with the human CMV. I know that the human CMV tried to copy also our own self-peptides, but I haven't seen that with EBV peptide from my library.
Okay. And Dr. Herbert, for the work that you showed, especially for your recent Science Immunology publication, one of the questions that came up was the expression of IL-33 and its general impression as an inflammatory cytokine, given it being an IL-1 family cytokine. But it seems that your work seems to propose divergent roles for IL-33, or potential alternative roles for IL-33 in the context of helminth infections. So do you see divergent? Does your work seem to back up that divergent role for IL-33? Is it not just an inflammatory cytokine?
Correct. So IL-33, so there's other members of the, let me kind of step all the way back. There are other members of the IL-1 superfamily that also have immunosuppressive functions, immunoregulatory roles. IL-36, 38, there's some less well-studied members. The most well-studied members are IL-1, alpha and beta, and IL-18, and those are, even those can, in certain contexts, there have been reports showing that they're not purely pro-inflammatory. IL-33, which is what we've been focusing on, because the receptor ST2 is widely expressed on a variety of different cell types, that already before we started the study kind of implied that different T cell lineages could use IL-33 for different purposes and not solely an inflammatory gene expression program. And most importantly, the T regulatory cells, and there's many different flavors of T regulatory cells. One flavor is that one that expresses ST2. So the premise was already out there, right? People just didn't understand at all, how could this one cytokine have an inflammatory role in some contexts and anti-inflammatory roles or suppressive role in other contexts. And what our paper does is it uses a pre-lock system to restrict IL-33 expression, or rather delete IL-33 expression in either epithelial cell lineage, which is the more canonical pathway, or the dendritic cells. And that was the basic observation, was if you delete in these two different major cell lineages, you get different outcomes. And not in this paper, but in another paper, we've also looked in this context of colitis. So it's not just parasitic helminth infections, that this different cell source has a different functional outcome. And the paper takes one further step by saying that the delivery of how you bring the eggs to the door, right, is also how those eggs are received. I don't know if the egg analogy really works there, but let's just go with it. In that epithelial cells make a lot of IL-33, and they generally, when they die, they will elaborate this into the extracellular space. Dendritic cells make quantitatively an order of magnitude less, right? So they make very small amounts of IL-33, but they have this really precise mechanism to deliver it specifically to the T cell at the time that the DC T cell conjugate is forming. I like to think about it in a way, liken it to desert farming. So desert farming, if you don't know, is like places like in Israel and other, very kind of water poor areas of the world. Irrigation is done by providing a small amount of water precisely at the root base. Whereas we're more familiar in the US with this kind of, large crop dusting kind of approach where volumes and volumes of water are spread over a large area. And so if you look at that analogy, which I'm sure does work, you can see how you can kind of be convinced that providing the water or here IL-33 in different contexts could really have a very different biological outcome, right? So yeah, and we find the protein that is doing that delivery. So it's pretty cool.
That's really cool. And Dr. Morrison, for you, the work that you presented on these plural macrophages was really fascinating. I was wondering, can you describe the phenotype of these plural macrophages? What makes them distinct from other macrophages? And have you incorporated any of the? We live in the time of lineage tracing mice as well as single cell transcriptomics. Have you been able to leverage any of those new emerging tools in hoping to try and better characterize these plural macrophages?
Okay, great questions. So the plural macrophages, specifically the small ones, they are MHC class 2 positive, F480 low, CD11B positive, and CD115 positive, and they express MERTK as all macrophages do. As far as single cell sequencing, that is definitely something that we want to do. The lab was locked down here in Riverside for due to COVID for quite a while, but we're back up and running and trying to get those kind of experiments done. So we definitely are interested in the single cell sequencing and seeing what kind of phenotype the macrophages take on once they enter the tissues. At this point, it looks like they're interstitial macrophages, but since there are multiple interstitial macrophage types, we haven't actually gotten to that step yet. And so we'll be employing microscopy, and we'd like to do some of that spatial profiling that's really hot right now. So it's a wide open field. This project is very, very new. I mean, I don't know why we're even sharing it with people, but I like the story so much that there's a lot. Need students, postdocs, whatever, to come on and help drive this forward.
Well, just to reassure you, we're here to amplify the amazing work everyone's doing. We appreciate you sharing that exciting work. One of the other questions that I wanted to open up to all of you is, how do you see your work bearing out on the current pandemic that we're in with COVID-19? I think all of you are doing fascinating work, and I see unique touch points, ways in which each of your stories might help shed light on our understanding of COVID-19. And I was curious to hear from each of you, how do you see your work helping to shed light on better understanding COVID-19?
I will start. I think it's pretty obvious for Juliet studying flu. I have a collaborator doing flu, but I'm not a viral immunologist. I think that, you know, probably the most exciting part was finding a delivery mechanism for a cytokine. And, you know, some of these COVID patients, it's been described a cytokine storm during a severe SARS-CoV-2 infection. Cytokine storm is something that's not limited to COVID. I mean, it's also a phenomenon in ARDS, sepsis, a whole host of other very severe burns. But it's when innate and adaptive immune cells actually start to elaborate volumes and volumes and volumes of cytokines, and leads to organ shutdown. And so, you know, what my lab is doing right now, very fervently, I might add, before we get scooped on this, is to develop inhibitors for this delivery mechanism to kind of block that cytokine delivery. And the idea would be, you know, develop a molecule that, a drug, small molecule inhibitor, you name it, antibody, that can block the pore, right? And so if you're undergoing acute respiratory distress syndrome, cytokines pouring out of all your immune cells, here's a stopper. So that's kind of how I'm thinking about it right now.
I can jump in. Well, for my work, I think how it can be applied to the COVID-19 research. So earlier during this pandemic, when everybody was kind of trying to publish and understand this disease, there were a lot of immune profiling that was done, and also research of potential antigen target. And they used those methods that we use for NK cell research as fundamental biologists, which is the identification of antigens that bind to either class 1 and class 2. And there is a list of peptides that came out that look interesting in terms of eliciting immune response. And you can see, and there were a different cohort that was compared in terms of responding to those viral peptides coming from SARS-CoV-2 virus. And you can see that people who were able to be the donor that were asymptomatic were actually the one that was having a good immune response, and it was mainly T-cell driven immune response. And the one that had severe symptoms were the one that was kind of failing on having a good cytotoxic immune response. Regarding NK cells, well, they were kind of MIA in this profiling because there were a huge decrease of them in the periphery. And yeah, so yeah, that's it for me.
Okay, I'll speak now. So as far as COVID-19 goes, I think the work that I'm doing with influenza is pretty transferable. And so I would be very interested in finding out whether SARS-CoV-2 infection also leads to this recruitment of these pleural macrophages. We finally actually have our biosafety level 3 facility set up at UC Riverside. So fingers crossed, in the new year, we can set up that experiment.
For me personally, so initially I'm trying to find out what IL-22 is doing in healthy pregnancies, but babies are still being born, COVID or not. But I tend to use uterine samples from women who are, I should say, healthy and otherwise. So if COVID is affecting women and somehow getting across the placenta into the lining of the womb, then I might expect that there might be an immune response there. And I'm not sure exactly if the ILC3s will then increase in number, because usually they are targeting they're targeting non-immune cells. So these are like the epithelial cell structure cells. So if the epithelial cells are damaged, then a whole plethora of immune cells might be affected, not just necessarily the ILC3. So in that respect, it would be interesting to see what happens in terms of COVID-wise in pregnancies at term. Usually when infections happen earlier on in pregnancy, that pregnancy tends to miscarry. And I'm not sure exactly how COVID will affect those earlier on. Yeah, so it's interesting. I'm not quite there yet. Let's find out what ILC3 and IL-22 is doing, first of all, in healthy pregnancies before I go on to that.
Well, with your work in mind, Antonia, one of the questions that emerged from our listeners was what ILC3 is doing. And so do you currently know if ILC3s are interacting with uterine epithelial cells or other immune cells to mediate their function, or what they might be doing when pregnancies are not successful?
So most of what we know, because we're only recently starting to understand the phenotypes of what the cells look like during different phases in pregnancy. Most of what we know comes from the first trimester pregnancies. And we know that ILC3 produces other cytokines known as IL-8 and JMCSF. So they can recruit and activate neutrophils during the first trimester, and this is thought to contribute to establishing the placenta. We know that the IL-22 is also produced by ILC3 during the stage, but we don't know exactly what it does. So one of the previous studies that I've read showed that decidual NK cells are able to recruit or promote the invasion of trophoblasts, but this is because they did not make a distinction between NK cells and ILC3, because ILC3 shares surface markers with both NK cells and also LTIs. This is why the recent developments in immunophenotyping are so important, because then you can really tease them apart individually. So now that we can do that, we can now be more specific about what we try to do to find out what do they do exactly, and which cells are contributing which cytokines to certain functions. So that's sort of where I'm starting off with as well in my PhD. Sorry, Dan, I think you're on mute. I can't quite hear.
Thank you. Bernice, one of the questions that emerged from our listeners was wondering, how do peptides affect the interaction with NKG2C, and do your peptides make a difference with the binding?
Something that I didn't actually went through during this presentation was that when we did the flow cytometry assays, either for NK cell activation or target cell killing, we had to exclude NKG2C because NKG2C also recognize HLA-E, and we didn't want to have that background noise when we looked at NKG2A responding cells. So for flow, we were able to basically analyze NKG2C negative, NKG2A positive cells. And when we did the killing assay, we basically had to sort NKG2A positive, NKG2C negative cells prior to the co-culture. So those are the data I didn't show, but they're all in the paper. You can see the grading strategies and how we picked out those cells. But that's a good question.
And Juliet, for your work, the pleural macrophages, are they similar to peritoneal macrophages that contribute to clot formation?
Yeah, they are actually. So the pleural macrophages and peritoneal macrophages are very, very similar. So both of these serosal cavities have these small macrophages as well as the large macrophages. And when you profile them, gene expression wise, they're pretty similar.
That's fascinating. And Dr. Herbert, for you, it seems that the really encouraging quote that you read at the end of your talk has excited a lot of people and encouraged a lot of people. There's already a lot of people excited about what you said on Twitter, and so I'm going to use this time to kind of transition from the science a little bit more to our personal experiences and kind of thinking about being Black in immuno. Can you tell us where that quote came from? Did you write it? And if you did write it, what inspired you to write those words?
Yeah, so that's my original work. I've used journaling for a very, very long time for survival, quite frankly. I'm not going to turn this into, what all my experiences were. I will say that it comes from deep within me, based on all those experiences, before grad school, during grad school, get invited to, start giving talks, and the way I was treated, and just, you can't take on everything can't be a fight, right? At the end of the day, I am a basic scientist, and I need to know that that's exactly who I am. The motivation behind writing that poem, I want it to be used. Hopefully my trainees, they seem to like it as well, because it's what I would have wanted to hear during my formative years, because there are some distinct challenges. There are additional challenges out there, and we're Homo sapiens, so we thrive off of each other. Like, we are creatures of community, right? And if your community is not providing you, or you're not identifying with the community in its totality, one can get lost in there with identity. So the purpose of that poem is to put a flag in the sand, in the soil, for like how I see myself today, and how, when the going gets tough, hopefully, some second year grad student preparing for prelims or something can read it and say, damn it, I'm going to do this. And, and even if I have to go and repeat my prelims, so what? I'm going to keep on trying until I succeed. And that was the purpose.
I think what was so powerful about your words was that not only did it give voice, I think, to the struggles and trials and obstacles that I think so many of us face in our respective fields. I think what was also powerful about it was that it was also affirming. It was also encouraging. Mm-hmm. It also, and just quickly hearing you read those words, it was very life-giving. Mm-hmm. And so one thing I want to ask of all of our panelists is what, as we navigate through our careers, what is something that has given life to all of you as you navigate being Black in immunology?
For me, navigating being Black in immunology, it's basically the same as navigating being Black anywhere, I think, in society. There is a lot of anti-Black sentiment that is pretty global, and it does impact, it makes it much harder to be your authentic self when you're always worrying that the people that you're interacting with are actually, seeing you as less than. The way I deal with that is by surrounding myself with colleagues like myself. Like, I have a whole bunch of people of all hues that I speak to about whatever microaggression or whatever. And so having a chat group and people I check in with regularly, it actually keeps me going. And also talking to my family, and they're really, really proud of me. So sometimes just getting that check and that feedback is enough to keep you just sort of like, yeah, wait a minute, of course, yeah, I am awesome. What the hell am I thinking because of this person's little snotty comment or this microaggression? Like, you have to, recenter yourself, remember why you're here, remember all the people who have your back and want you here, and then just brush it off. That's how I do it. And I just have no problem just cutting people out, even if they're super famous or, should have some pull or some sway in my life. If you're if you're not a nice person, I don't want you around me anyway. And so I will not be reaching out to people who I think are misogynist or who are racist. I don't want to collaborate with you. And I don't feel I don't feel bad about it. I'm not going to compromise my own sort of view of the world and how people should be treated just because somebody's a big shot or something like that. So, I mean, it does lead you, of course, to, therefore, there are less opportunities. But for me, I think, you know, are you really burning a bridge if the bridge leads nowhere, right? So there's that, yeah.
Yeah, I'll jump on and second what Dr. Morrison just said. I think surrounding yourself with people who support you, cheer you up, it's really important into, you know, navigating this in science. Having good friends, I have a good surrounding of friends here, and also my family is really supportive of me, and I think they're watching right now in France. Also I want to give a big shout out to the Stanford Black Immunology the Stanford Black Postdoc Association at Stanford, really supportive group of strong female and male, and they are really, they made a difference into my experience at Stanford, and I'll be forever thankful for them. Yeah, so I think, as Dr. Morrison said, the surrounding is really important, and where you do science and with who you do science.
I'll jump in then after. So I agree with Bernice that where you do science and with whom is very important. You want to choose mentors or PIs or a team that you can actually work with. And that's important so that you can feel at least that you're making progress in your own career, or if it's just something that you just want to keep doing and not make a career of it, you want to be able to go in every day and be okay at the end of it and go home. Also, family is a big thing for me, and they keep me or remind me of who I am, even so that's quite helpful, because you sometimes find yourself in spaces where people may not recognize, you know, your culture, you as you are. They may not accept you as you are, as a scientist, as a woman, as a Black person, altogether. So yeah, wherever you can flourish, that's where you should go. If it really costs you way too much to be there, then that's the time when you start thinking you've got to move.
So it seems that, you know, for almost everyone, it seems that the dynamic of the people around us is really critical. And oftentimes in immunology, the people around us, very few of them are Black. Very few of them have the same experiences, same views. But one of the ways to bridge that can sometimes be finding an ally, finding the supportive people that might not share the same experiences, but still affirm and support us. And so what has true allyship looked like in each of your experiences?
Okay, so true allyship. For me, I think it's the people who can see a microaggression and talk and speak up about it. I find that a lot of people are very, I don't know, complacent, uncomfortable when they, you know, like if somebody utters something racist or sexist, they might they'll just keep quiet. But I think a real ally is one who says, really, you said that? Come on, right? Like somebody who can call out their own peers and not leave it to the person who is being, harassed or, demeaned. So I think people who I see as true allies are the people who are able and confident and able to do this. And also where it's not just performative. If you are a true ally and you do think that Black Lives Matter and Black science matters, are you citing Black scientists? Are you inviting them to come speak? Are you treating your Black trainees the same way you treat your white trainees? Who gets to come over for dinner? Is it just one group of people versus another? That to me is a true ally is someone who actually checks themselves. And because we all have grown up in this society that does have this slant, this anti-Blackness built into it. So we all have to check ourselves, even us as Black people. We hear the messages every day. So it's really hard not to just be completely taken over by imposter syndrome. And so the best allies out there are the ones who also recognize that that anti-racism is an active process. And so those are the people who are out there mentoring Black folk and people of color, and who are inviting them to speak and giving them the sort of opportunities that we see being promoted for our lighter colleagues.
Yes.
Yeah. Go ahead. Oh, sorry.
Sorry,
go ahead.
Antonia. No, go ahead.
Okay. Am I on? Yes, I am on. I agree with what Juliet said, Dr. Morrison said. Yeah, a strong ally does amplify their Black colleague. They do treat them as well in their training or supervision as they do their non-Black colleague or non-Black trainees. I think I also like the point about also being able to check yourself. That's something that not everyone has grown up with, and that has to be learned. And that also is like a personal responsibility. So you can't tell other people to do it, but you hope that folks will, including yourself. So yeah, strong. Yeah, I agreed with those points. Strong allyship is being really honest about yourself, with yourself, about promoting Black people, inviting Black immunologists to seminars, giving them that opportunity to share their work. And I guess even before this point, it's like the procedures that allow them to even get to that point as well, being fair in those procedures, because it's taken my journey has been a bit of a meandering to get to a PhD. So yeah, I think one of the reasons that was because I didn't have that insight or that that person that I knew before to discuss that path. And even though you'd make attempts at getting insight and getting to know folks, I think the networking scenarios that I found myself in usually weren't as welcoming sometimes. And so trying to develop those networks are quite difficult if for example, you're just considered sort of not welcome in the first place. So the pathways as well, being fair before you get to this point, is also something to consider and be fair in. Even if you're not a senior person, just lay colleagues that you're working with, just be nice in research would be a good thing. Yeah, that's me.
Yeah, so I think you both have said it all. And if I have to add a little bit to this amazing words, I think you find your allies with people with whom you can be really yourself as a Black trainee. And I think being able to express yourself without feeling judged or put into a box, it's really important as well. So, yeah.
Of the key relationships I think we have in science, regardless if it's academia, the clinic, industry, mentorship is a critical relationship. A mentor is a critical relationship where allyship can have the most good, or the absence of it can be incredibly detrimental. Can each of you share from your own personal experience ways in which mentors have served as incredible allies? Or if you didn't have that, what are ways that you wish a mentor did serve as an ally?
I can start. So I've done research in France, in England, where I did my PhD, and I'm doing a postdoc here in America. And I have to say, I've been pretty lucky to find pretty good mentors in terms of allyship. And they always push me to be able to go to conferences, to have fellowships, and to promote my work. And I will be always grateful for that. Yeah.
Yeah, I agree with Bernice. The best of the mentors are the ones who are constantly pushing you and who want you to actually get that exposure. So to, to go to conferences, meet people, take ownership of your work. That's the definition, I think, of like a really good mentor. On the other hand, if one should worry if your PI is kind of keeping you away from everything and keeping you away from presenting and doing all of these things that are absolutely critical for your career. So I think, yeah, that's what a real mentor does. And it's also a person who makes sure that you are meeting your goals, right? Like it's so easy to kind of get off track, especially in a PhD. And so having a person who is accountable and who actually cares that you are going to, you'll finish this and finish successfully is important.
Yes, I agree with all of those. Those are very good points. Yeah, having a mentor who has your best career interests in every decision and meeting that you have together is very helpful and important. And it also makes you feel like you have the support, the background to finish the race that you're in, the PhD, the project, get the grant. I'm fortunate enough to have that right now, so that's quite good. And yeah, it would be nice if all students had that. So yeah.
Before I forget to mention it, unfortunately, Dr. Herbert had another commitment, but I wanted to say we thank him for his contribution. We thank him for the work that he's doing, the work that he shared with us today. We have a couple questions left. I next wanted to hear from each of you if you can share how you think that we can improve our connections with our communities and to help try and rebuild trust in science in this critical moment.
That's a major one. I think it comes down to effective science communication. So far, most scientists do not really talk about their research in an accessible way. And it makes it very easy for people who are hell-bent on misinforming people and manipulating people. We leave this vacuum open for them to fill it. So I think the best way to get people on board with scientists is to make what you're doing pretty accessible. I mean, if you can explain your research to your non-science colleagues, you can certainly explain it to the general public. And when people see that level of openness, they are much more likely to actually trust in what you're saying. Like if you bring it to their level and you emphasize the important parts. I think it also helps that we should have we need more Black Black scientists and scientists of color in general and in healthcare, because our population, the makeup of science and academia and all of this should reflect the makeup of the population. And so there's a lot of mistrust, well-earned mistrust actually, in the Black community when it comes to listening to what these scientists are saying or which public health officials are saying. And it's for good reason, because for many years and even to this day, there are glaring inequities in outcomes. And I think that can only really be addressed by actually having more people at the table. We need more Black professionals, and we need more people who actively care about our communities doing this sort of work.
So I would like to add that doing some outreach and also mentorship is important. as postdoc or even PIs, it's nice to mentor people at really the beginning of their training. Like, for instance, people transitioning from high school to universities, or within the STEM program as well. So I really value the different outreach programs that are put in place, especially in my institution, Stanford, to be able to have students on campus and teach them science and how, actually how the lab can be accessible to them, and show them their potential.
Yes, I agree with both your points. Opening up that communication with the local community is important, whether it's starting out as young, even as primary school and secondary school students, which is like, what do you call it, elementary and high school for you guys? As well as explaining your science or your research in a more accessible way to the general public. They'll feel less afraid, because it's something that it doesn't seem so foreign anymore. It seems more practical and makes sense. And it would help if more Black scientists and healthcare workers were involved, but not just on a frontal show, but with real impact would be more beneficial, and that's quite visible. So yeah, moving forward, I think that would help the community to engage what scientists do and science in general.
For our final question, I would like each of you to tell us, what does being Black in immuno mean to you?
For me, it means a lot. And I was extremely thrilled when I heard that Black Immuno Week was happening. And I wanted to jump in and participate to this week, and also the following actions that will come up with this organization. I always hear that people can be the change that they want to see. And I feel like, as a Black trainee, a Black scientist, I kind of like to put effort to things that I feel like need to, how to say this, put effort in terms of having, making a change, basically. And I really thank Black in Immuno for starting this, and giving me the opportunity to participate and showcase Black immunologists. Thank you.
Yes, for me, being Black in immuno, or actually hearing and joining you guys, is really a really big honor. Thank you so much for doing this. This feels like a dream come true a little bit, because yeah, you do sometimes, as you mentioned, Bernice, that you're often in spaces where there are not that many of you, and now it feels like, okay, you guys were always there. I was just busy either doing research or crying because an experiment didn't work, but it's okay, or yeah, other things. So it's nice to have this network now exists, and also it's nice to be able to share you guys with the young people sometimes that I encounter, saying that I'm not the only one. It's like my niece, as I heard her school, she says her mom says not many kids in her class can say that their aunt is a scientist. So it's like, okay, that made me feel really proud, and you guys made me just feel like you just amplified that effect 100 times more. So yes, thank you very much. I feel honored, and I know this is things that we can do anyway, but it's just nice to see you there and to see you show your work and see you guys in senior positions. So thank you.
I completely agree. It feels really good to be a part of this. I am so excited for this whole program that you've put on. The organizers, thank you so much for inviting me. Like, it is an honor. And I'm so excited to see all of these faces that I didn't even know existed out there. Sometimes you are the only person in the room, and it just feels, I don't know, it lifts my spirits to see this, and then to see all of the excellent science and that is being done by Black scientists across the world. It's really a nice feeling. And I hope we can continue to do things like this, because it makes me feel a lot more a part of a community than I felt previously.
I couldn't have put it better myself. Thank you all for your, thank you all so much for sharing your experiences, your insights, your wisdom, and the work that you do. I loved it. This was great. Thank you so much. For our listeners, thank you for tuning in. That concludes the TEDx talks for innate immunity. Dr. Morrison, Dr. Imberindi, Dr. Herbert, and Antonia, thank you all so much for sharing your work. And I wish you all well. And for our listeners, keep tuning in to the rest of Black